Looking at the transport across the cell surface membrane. The phospholipid bilayer creates an effective barrier against water-soluble molecules and ions. It prevents aqueous contents from escaping. However, some exchange between the cell and its environment is essential. To achieve this, diffusion, facilitated diffusion, osmosis, and active transport, as well as bulk transport, will be needed to help move those substances across the membrane. Suggest 3 reasons why exchange between the cell and its environment is essential. Go ahead and jot these down in your notes and we will discuss them during class. Diffusion refers to the net movement, the result of random motion of molecules or ions. With diffusion, the particles are going to go from an area of high concentration. Know there's more particles on the left side than there are on the right side. Substances are going to diffuse from high concentration to a low concentration. This is what's known as a movement down the concentration gradient. It's a net movement because particles can move back and forth. Notice your arrow is longer going towards the right, indicating that your particles are going to go from high concentration to low concentration. There can be some movement returning back, but notice it's a much shorter arrow, very little. This process continues until the particles reach an equilibrium. You're familiar with diffusion at home when somebody's baking or making popcorn and you're in a different room and all of a sudden you start to smell it. That's diffusion of the particles leaving the kitchen and going to the rest of the house. It's due to the energy that's within the particles. Energy is not required for the particles to diffuse, to go from an area of high concentration to low concentration, but the particles themselves have energy in them. This is how oxygen and carbon dioxide are able to move across the membrane through diffusion. Both oxygen and carbon dioxide are uncharged and nonpolar. There are factors that can affect the rate of diffusion. The steepness of concentration gradient, referring to a greater number of particles. If there's a higher number of particles, greater concentration, they're going to diffuse faster. Temperature, higher temperatures allow for a higher rate of diffusion. Surface area, the greater the surface area, again, more diffusion is able to occur. And then the nature of the molecules or ions, small molecules are able to move at a greater rate than larger molecules. And remember, if it's a hydrophobic molecule, they can also move across the membrane because the interior of the membrane is hydrophobic. Taking a look at steepness of concentration gradient, again, when you have a greater concentration, the particles are able to diffuse at a faster rate. So when we look at our 2 diagrams, notice in this top one, we have a faster rate of diffusion. You're looking at the red particles diffusing across the barrier. There's more red particles here, therefore they're going to diffuse faster. In the second diagram you're looking at, there's a few red particles already on the right side, and then not nearly as many on the left side, but it's still a higher concentration on the left versus the right. So it's going to diffuse, but it's going to diffuse at a slower rate. Taking a look at temperature, we have 3 different beakers with water in them at different temperatures, 20, 65, and 90 degrees Celsius. Higher temperatures are going to have more kinetic energy. Therefore, the particles are going to diffuse faster, and this is shown with the little red cube placed in each one of the beakers. The one at 90 degrees Celsius definitely has a much darker color of red than the one at 20 degrees Celsius. Taking a look at the nature of molecules or ions, larger molecules require more energy to get them moving than small ones. Larger molecules tend to diffuse slower than small molecules. Remember, diffusion itself does not require energy. Molecules and ions will simply go from high concentration to low concentration. This large molecules require more energy, referring to the energy that the molecule or ions have themselves. The larger they are, the more energy they need to make themselves move. Nonpolar molecules such as glycerol, alcohol, and steroid hormones diffuse easier than polar ones. The nonpolar molecules are soluble in the nonpolar phospholipid tails. The greater the area, the more molecules or ions that can cross the membrane, so a faster diffusion. The surface area of a cell's surface membrane can be increased by foldings, such as the microvilli in the cell's lining of the intestines or the cristae inside the mitochondria. An important point is that the surface area to volume ratio gets smaller as the cell gets larger. Taking a look at how that's possible, we're going to imagine that these cubes represent our cells. The larger the cell, the smaller the surface area in relation to the volume. Surface area for a cube is 6s squared. Volume is length times width times height. Something biologists are concerned with is the surface area to volume ratio, which is surface area per volume. So if we found the surface area for our small cube, meaning our smaller cell here, we get 24 micrometers squared. The volume for the small one is going to be 8 micrometers cubed. Taking a look at the surface area to volume ratio, that's 24 to 8, which is going to simplify down to a 3-to-1 ratio. Then you'll notice as you look at the larger cube or larger cell, it got larger, but then our ratio is smaller. Go through and find the surface area and volume. You get a 96 to 64 ratio, which reduces to a 1.5 to 1 ratio. So you'll notice how the surface area per volume got smaller as the cell or the cube got larger. Cells rely on diffusion. Diffusion is only effective over a very short distance. Most eukaryotic cells are no larger than about 50 micrometers in diameter, and prokaryotic cells are even smaller. An aerobic cell would quickly run out of oxygen and die if it were too large. Question time. The fact that surface area to volume ratio decreases with increasing size is also true for whole organisms. Explain the relevance of this for transport system within organisms. Jot down your information and we will discuss this in class. Respiratory gases can cross the membrane by diffusion. Uncharged and nonpolar particles can cross the membrane by diffusion. Water is polar, but it is able to cross the membrane because it's such a small molecule. So now we're going to take a look at how large polar molecules are able to cross the membrane. Large polar molecules, such as glucose and amino acids, or sodium and chlorine ions, cannot diffuse through the phospholipid bilayer without the help of proteins. Facilitated diffusion takes place with the help of certain protein molecules. The protein molecules are channel proteins or carrier proteins. Channel proteins, proteins with water-filled pores. They allow for charged substances, usually ions, to diffuse through the membrane. The pores are gated, meaning they can open and close to be selective when those materials are going to pass through. Two examples for gated proteins found in neuron cell surface membrane would be the control of sodium and potassium ions. In this first picture, it's representing where the gate is closed, the ions are not able to move through, and then they receive a signal that opens up the gate, and so the sodium and potassium ions can move back through the cell. Here's a second one showing you again the gate's closed, the sodium ion is not able to move through, receives that signal, allowing it to open so the sodium can move through. There are channel proteins that require energy, but those would fall under active transport, and active transport moves against a concentration gradient. We'll get into this in more detail later, but the sodium ions are going to move across the channel protein, and then they have to be restored back to the outside of the cell, and that's done through active transport. Carrier proteins can flip between shapes to alternate their opening binding sites on the interior or exterior of the membrane. This is going to depend on the concentration gradient inside and outside the cell. You have a higher concentration here, so the protein is going to open, allows for the substance to enter, and then it would close and then open on the other side to release the substance. Carrier proteins that are known as pumps require energy. That would not be an example of a facilitated diffusion. That would be active transport because it requires energy. Remember, diffusion and facilitated diffusion are without energy. The direction of movement depends on the concentration. Substances move down a concentration gradient, meaning they move from a high concentration to a low concentration. The rate of facilitated diffusion is affected by how many channels or carrier protein molecules there are in the membrane. Channel proteins also affect the rate by having the pores open or closed. Channel proteins keep their shape, and carrier proteins can change their shape. Okay, osmosis is a type of diffusion, but it is only going to involve water molecules. When we talk about water diffusing or water going through osmosis, again, we're going from an area where there's a higher concentration of water to an area where there is lower. However, we don't say high concentration to low concentration. We would say the water potential. This has a higher water potential. In other words, there are more water molecules. So in a sense, it is a higher concentration, but we need to change our vocabulary, referring more to the water potential. There's more water potential on the left. Therefore, the water is going to go through osmosis, move across our semipermeable membrane to the area where there's a lower potential of water. Looking at water potential, it's the tendency of water molecules to move from one place to another. It's measured by the Greek symbol psi. You will also see kilopascals when you're measuring the pressure. When we look on the left side, the little white dots are representing the water molecules, and then the darker gray are representing the solute. So the left side has a higher water potential, meaning there's more water molecules here, and less solute. So the water molecules, again, are going to go from a high water potential to an area of low water potential. Water potential for pure water is 0, meaning it doesn't have any solutes dissolved within it. Solutes are going to make the water potential less than 0. The more solute, the more negative the water potential. When you add solutes such as sugar being dissolved in water, you're lowering that water potential because now you've got solutes where water molecules. You've lowered the water potential, so it's becoming a negative value. The amount that solutes lower the water potential is what's known as a solute potential. Applying pressure to solution B causes some of the water to be forced to go back into solution A. By increasing the pressure on B, you're increasing the water potential to move from B into A. That is an increase in water potential until it's higher than the water potential in A. Pressure on a solution increases its water potential. Pressure potential. Increasing pressure increases water potential. The contribution made by pressure to water potential is called pressure potential, and it's given the symbol psi with a little subscript p, meaning you're measuring the water potential due to pressure. Pressure potential makes water potential less negative, meaning more positive. Question time. In figure B, the system has come into equilibrium, so there is no net movement of water molecules. What can you say about the water potentials of the two solutions in A and B? Please jot down your thoughts and we'll discuss it in class. In figure B, the solutions A and B are at equilibrium. Which solution, A or B, is more concentrated? Why does water not move from the more dilute solution to the more concentrated solution? Taking a look at water potential in animal cells. These are representing red blood cells that are placed in distilled water. Means water is going to move into the cell swells and burst. If red blood cells were placed in a concentrated salt solution, water is going to leave the cells and the cells shrink and shrivel. Meaning if the water potential of the solution surrounding the cell is too high, the cell swells and bursts. If it is too low, the cell is going to shrink. Question time. Which solution has the highest water potential? In which solution is the water potential of the red cell the same as that of the solution? Osmosis in plant cells. Plant cells are surrounded by a cell wall which is strong and rigid. A plant that is placed in pure water or a dilute solution, if the water of the solution has a higher water potential than the plant cell, the water would enter the cell. The cell volume would increase, but the cell wall resists the expansion of the protoplast. Pressure would build in the cells, which increase the water potential of the cell until the water potential inside the cell equals the water potential outside the cell. In other words, equilibrium would be reached. The cell wall prevents the cell from bursting. A plant cell that is fully inflated with water is described as turgid. Looking at osmosis in plant cells, when plant cells are placed in hypotonic solutions, the water moves into the cell. But unlike an animal cell, remember animal cells in a hypotonic solution, they would take in so much water that the cell would burst. Plant cells won't burst because the cell wall is going to protect them. This is what's known as a turgor pressure in plant cells. Isotonic solutions, water moves into the plant as well as moving out of the plant cell. This is known as a flaccid plant cell. And then in a hypertonic solution, water leaves the plant cell, but again, it's going to keep its shape because of the rigid cell wall. What happens is plasmolysis occurs where the water is leaving the cell and it causes the cytoplasm to shrink. In other words, as it's shrinking, it pulls away from the cell wall. Question time. The two figures show plasmolysis. Why can plasmolysis not take place in an animal cell? A few more questions. If you would please pause the video. Make sure that you guys go through and answer these questions. And another question for you. Let's take a look at active transport. Active transport moves molecules or ions against a concentration gradient, meaning from a low concentration to a high concentration. This is achieved by carrier proteins called pumps. They are specific structures for particular ions or molecules. Requires ATP, which is adenosine triphosphate, for energy. ATP is produced during respiration inside the cell. The energy is used to make the carrier protein change its shape while transferring the molecules or ions. This can occur in or out of the cell. An example of a carrier protein that's used for active transport is the sodium-potassium pump. It's found in the cell surface membrane of all animals. They use on average about 30% of the cell's energy, but in a nerve cell they use about 70% of the cell's energy. The role of a sodium-potassium pump is to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. Both sodium and potassium carry a positive charge. Therefore, the net result, the inside of the cell is going to become more negative than the outside. The difference in charges between inside the membrane and outside creates a potential difference, which allows nerve cells to propagate a signal. We will discuss this potential difference in a much later chapter. The red area indicates a receptor site for ATP. The receptor site, ATPase enzyme, is bringing about the hydrolysis of ATP to ADP. Hydrolysis is the breaking down, so ATP is going to lose a phosphate group and release energy to become ADP. Active transport can be defined as the energy-consuming transport of molecules or ions across a membrane against a concentration gradient. Two areas where active transport is important. The reabsorption in the kidneys, where certain useful molecules or ions have to be reabsorbed into the blood after filtration into the kidney tubules. And then active transport is involved in the absorption of products in the digestion in the gut. Looking at plants, active transport is used to load sugar from photosynthesizing cells in the leaves into the phloem tissue for transport around the plant. Bulk transport. Bulk transport involves the mechanism of moving large quantities of molecules into the cell, known as endocytosis, or out of the cell, known as exocytosis. Large molecules such as proteins or polysaccharides, parts of cells, or even whole cells may be transported across the membrane. Endocytosis involves the engulfing of material by the cell surface membrane to form a small sac or endocyclic vacuole or vesicule. There are two forms, phagocytosis and pinocytosis. Phagocytosis, or cell eating, this is bulk uptake of solid material. Cells specializing in this are called phagocytes. The process is called phagocytosis, and the vacuoles are called phagocytic vacuoles. An example is engulfing of bacteria by white blood cells. Pinocytosis, or cell drinking, this is the bulk uptake of liquids. The vacuoles or vesicles formed are often extremely small, in which case the process is called micropinocytosis. Human egg cells take up nutrients by pinocytosis before fertilization. Exocytosis is the opposite of endocytosis. Exocytosis is a process by which bulk material is removed from the cell. Example, how plants transport materials out of the cell surface membrane to make the cell wall. Secretion of digestive enzymes from the pancreas, secretory vesicles from the Golgi apparatus carry the enzymes to the cell surface membrane and release their contents. One last question. There are more mitochondria than average in pancreatic acinar cells. Suggest a reason for this. Pancreatic acinar cells are a highly specialized structure developed for synthesis, storage, and secretion of digestive enzymes.